We've made enormous progress over recent decades in understanding how tumours grow and develop, and we've developed new targeted treatments that are having a real impact on survival. But for many patients, including patients with diseases such as multiple myeloma or breast or prostate cancer, the disease can relapse, and it often relapses in distant sites. And one of the sites that most commonly experiences relapsing disease is the skeleton. And one of the things that we've been interested in is trying to understand why cancers relapse in these distant sites. Because once they are in those distant sites, they become notoriously difficult to treat. One of the things that we've discovered is that cancer cells can spread from the primary tumour to these distant sites, and they sit in these sites and can reside there in a long-term dormant state. And then something wakes those cells up, and once those cells have been woken up, they give rise to new cancers and new disease. So one of the challenges in the field has been an ability to actually find dormant cancer cells. So we were one of the first laboratories to really develop technology that would firstly allow us to identify a dormant tumour cell and then be able to find that dormant tumour cell deep inside the skeleton. And we've had to bring together different technologies. So one of the exciting things using these new technologies is it gives us an ability to see a cell as it arrives in the skeleton for the very first time, and we can see where it localizes into the skeleton. And what we've discovered is they migrate to very specific locations within bone, and once they engage in those environments in the skeleton, they switch on a whole new repertoire of genes that we had no idea were involved in controlling the behavior of cancer cells for the very first time. And we can use this new understanding, this new knowledge about the genes that control dormancy to start to think about how we can target those cells in order to eradicate them and remove them so they don't give rise to relapse in the future. So our aspiration is really to think about new treatments that target relapse before relapse ever starts. But what we would like to do is couple new targeted treatments for primary disease with new dormancy targeted treatments. So we can tell a patient when they come into the clinic, firstly, that whether they're likely to relapse later on, and if they are, offer them new treatments that are coupled with their primary disease treatment to stop relapse happening in the future. We're at the very beginning of an opportunity to really think about an ability to cure a cancer. And for many patients that do have these devastating cancers, they live with a constant threat of this returning. And this provides an opportunity to think about stopping that for the first time.